Good day, everyone. I am Lizelle Marie Ras from STEM Club St. Patrick of Ireland, and I will be discussing about the process of immune system in animals. So, immune system helps to prevent pathogens from gaining entrance to the body. Example, an outer covering such as a skin or shell blocked entry by many pathogens. Sealing off the entire body surface is impossible. It's because of gas exchange, nutrition, and reproduction requires opening to the environment. Immune cells produce receptor molecules that bind specifically to molecules from foreign cells or viruses and activate defense response. The specific binding of immune receptors to foreign molecule is a type of molecular recognition, and it is the central event in identifying non-self molecules, particles, and cells. So there are two types of molecular recognition: the innate immunity, which is common to all animals, and adaptive immunity, which is found only in vertebrates. Adaptive immune system response is also known as the acquired immune response. Innate immunity, which is includes barriers, defense, molecular recognition, relies on small set of receptors, protein that bind to molecules or structure that are absent from animal bodies but common to group of viruses, bacteria, or other pathogens. Adaptive immunity, which is molecular recognition, relies on a vast arsenal of receptors, each of which recognize a feature typically which found only on a particular part of a particular molecule in a particular pathogen. As a result, recognition and response in adaptive immunity occur with remarkable specify. Innate immunity. Innate immunity is found in all animals as well in plants. Two types of innate immunity. First, the innate immunity of invertebrates, which is repel and fight infection with only this type of immunity, and the innate immunity of vertebrates, which serves both an immediate defense against the infection and as the foundation for adaptive immune defense. Innate immunity of invertebrates is composed of largely and polysaccharide chitin. The exoskeleton provides the effective barrier defense against most pathogen. Chitin is also lined with the insect intestine where it blocks infection by many pathogens ingested with the food. Lysosome is an enzyme that breaks down bacterial cell wall further protects the insect digestive system. Innate immunity of vertebrate defense coexists with more recently evolved system of adaptive immunity. Barrier defense. Barrier defense of mammals which block the entry of many pathogens include the mucous membrane and skin. The mucous membrane that line the digestive, respiratory, urinary, and reproductive tracts produce mucus. Mucus is a viscous fluid that traps pathogen and other particles. The cellular innate defense. In mammal, as in insects, there are innate immune cells dedicated to detecting, devouring, and destroying invading pathogens. The toll-like receptor, or TLR, a mammalian recognition protein similar to a toll protein of insects. TLR proteins produce signals that innate response tune to invading microorganisms. Two main types of phagocytic cells: the neutrophils, which circulate the body and are attracted by signals from infective tissues and then engulf and destroy the infecting pathogens, and the macrophage, also known as the big eaters, which is the larger phagocytic cells. Some migrate throughout the body, whereas others reside permanently in organs and tissue where they are likely encounter pathogens. Example, some macrophages are located in spleen where pathogens in blood are often trapped. So there are two types of cells: the dendritic cells and the eosinophils also have roles in the innate defense. Dendritic cells, which mainly populate tissue such as skin that contact the environment, and the eosinophil, which often found the tissue in underlying an epithelium, are important in defending against the multicellular invaders such as the parasitic worms. Cellular innate defense in vertebrates also involve natural killer cell, which circulate through the body and detect the abnormal arrays of surface proteins characteristics of some virus infective and cancerous cells. Natural killer cells do not engulf stricken cells, instead they are release chemicals that lead cells to death. Adaptive immunity. The adaptive immunity response relies on T cells and B cells, which are type of white blood cells called lymphocytes. Lymphocytes in the third type remain in the blood and become the natural killer cells active in the innate immunity. Lymphocytes originate from the stem cells in bone marrow. Some migrate from the bone marrow to thymus. Thymus is an organ in the thoracic cavity above the heart. These lymphocytes mature into T cells. Lymphocytes that remain mature in the bone marrow develop as B cells. Antigens. Any substance that elicits a B or T cells response is called antigens. Antigens are usually large foreign molecule, either proteins or polysaccharides. In adaptive immunity, the recognition occurs when a B or T cells bind to an antigen such as bacterial or viral protein called antigen receptor. Antigen recognition by B and T cells. This antigen B receptors is Y-shaped protein consisting of polypeptide chains. Each light chains or heavy chains has constants region where amino acid sequence vary little among the receptor on different cells. The constant region of heavy chains contain transmembrane region which anchors to the receptors in the cell plasma membrane. Binding a B cell antigen receptor and an antigen is an early step B cells activation leading to formation of cell secret of soluble from receptor. The T cell, the antigen receptor consists of two different polypeptide chains, an A chain and B chain, linked by the disulfide bridge. Near the base of T cell antigen receptor is transmembrane region that anchors the molecule. The variable regions of the chain and together form a single antigen binding site. The antigen receptor of B cell bind to epitopes of intact antigens, purging from pathogens and circulating free in the body fluids. Antigen receptors of T cells bind only fragment of antigens that are displayed or presented on the surface host cell. The host protein that displays the antigen fragments on the cell surface is called the major histocompatibility complex or MHC molecule. So there are four major characteristics of adaptive immunity. First is the immense repertoire, which lymphocytes and receptors enable detection of antigens and pathogens never before encountered. The adaptive immunity, which is normally has self-tolerance and lack of reactivity against the animal's own molecules and cells. Next is the cell proliferation, which triggered by activation greatly increased by the number of B and T cells specific for an antigens. And last is the immunological memory, which means there are stronger and more rapid response to an antigen counted previously. And that's all. Thank you.